हॅलो स्टुडंट्स, आपण लास्ट लेक्चरमध्ये ह्युमन डायजेस्टिव्ह सिस्टीम ह्युमन डायजेस्टिव्ह सिस्टीम बघितलेली आहे आणि आता या लेक्चरला आपण फिजियोलॉजी ऑफ डायजेशन हा पार्ट बघणार. इन फिजियोलॉजी ऑफ डायजेशन we know that digestion is a process in which complex food material is converted into simple form, or the material which is undiffusible or unabsorbable, it is converted into diffusible and absorbable form. This digestion is a process in which physical and chemical changes takes place, and it helps in converting complex food material into simple form. In this digestion, physical and mechanical digestion takes place. In physical digestion, larger sized food particles are converted into smaller pieces. While in chemical digestion, here different enzymes helps to convert the small molecules into, large molecules into small molecules. Digestion takes place in different organs. Digestion takes place in buccal cavity, stomach, and intestine. First, we see here digestion in buccal cavity. In buccal cavity, mastication of food takes place. We know that in buccal cavity, there are different salivary glands are present. 3 salivary glands are present. These salivary glands secretes saliva. In this saliva, mucin and ptyalin is present. Ptyalin, it is salivary amylase. Here, mucus lubricates food, and ptyalin or salivary amylase convert starch into simple sugar, and this simple sugar is known as maltose. This is the reaction. Starch, that is polysaccharide, it is polysaccharide. It is also known as carbohydrate. Polysaccharide number of molecules come together and it forms here starch. Here salivary amylase acts on starch and it is converted into maltose. This maltose is disaccharide. This conversion takes place in the pH 6.8, that is slightly low. Next, digestion. This digestion takes place in stomach. All these processes you are seen in previous standard. Here it is the only revision. In digestion of stomach मध्ये काय होते आता बघा. In stomach, gastric juice is present. Gastric glands secrete gastric juice. This gastric juice, it contains HCl, that is hydrochloric acid, pepsinogen and renin. HCl, which is present in gastric juice, it functions, its first function, it kills bacteria. And HCl, it is acid, it provides acidic media for digestion or for a reaction. Then in gastric juice, pepsinogen is present. Pepsinogen is present in inactive form. This pepsinogen is converted into pepsin due to this acidic media. And another enzyme is also present, it is known as renin. Now, how HCl acts? We look here, HCl, it is present in gastric juice. Its functions are, it kills bacteria, it provides acidic medium, then it stops action of salivary amylase. Salivary amylase, which is secreted in buccal cavity. Then it converts pepsinogen to pepsin. These are the functions of HCl. Now, this pepsinogen, which is converted in the pepsin in the presence of HCl. and now this pepsin it acts on protein. So in the stomach here digestion of protein start. Proteins are digested in presence of enzyme pepsin and in acidic medium. Proteins are converted into proteases and peptides. This is the action of pepsin. Then third enzyme is secreted. This third enzyme is known as renin. This renin is only found in gastric juice of infants. The protein known as casein is present in milk. Renin is useful for conversion of this protein, that is it digests only casein, and this casein it is absent in adult persons. Then in gastric juice, gastric walls also secrete their mucus. This mucus is helpful. It prevents the action of hydrochloric acid on the wall of stomach because it layers the stomach. It gives layer, and due to this HCl from action of HCl stomach wall is prevented. This is partial digestion of food in stomach, and such a digested part is known as chyme. Here question is asked where chyme is formed, or after digestion in stomach the bolus is formed and it is known as, याच्यावर MCQ विचारला जातो, it is chyme. Then next digestion in intestine. Intestinal digestion it is very complicated. आपण इंटेस्टायनल डायजेस्टिव्ह सिस्टीम बघितलेली आहे. डायजेस्टिव्ह सिस्टीममध्ये स्टमक झाल्यानंतर इंटेस्टाईन सुरू होते. इन इंटेस्टाईन डिफरंट substances, different juices comes. In the small intestine, intestinal juice present, bile juice present, and pancreatic juice is also present. First is the action of bile. Bile is secreted by liver and it is stored in gallbladder. Bile contains bile pigments and bile salts. Now, how these bile pigments are produced? We know that lifespan of RBC is 120 days. After 120 days, these RBC goes in liver, and in liver from this bile pigments are produced, and the bile pigments are helpful for it gives color to fecal matter. Then another juice is known as pancreatic juice. Pancreatic juice it is secreted by pancreatic gland. This pancreatic gland it is leaf-like gland. It is mixed gland, exocrine as well as endocrine. Now this pancreatic juice it also secretes different enzymes. First enzyme is known as amylase. This amylase is known as pancreatic amylase. When amylase secreted in buccal cavity by salivary gland, then it is known as salivary amylase. But this amylase is secreted by a pancreatic. It is present in pancreatic juice and it is known as amylase. This is a pancreatic amylase. This pancreatic amylase acts on starch, and this starch is converted into maltose in the presence of pancreatic amylase. Here, bile juice provides alkaline medium. Bile is alkaline in nature. Other enzyme is secreted. This enzyme is known as pancreatic lipase. This pancreatic lipase, it acts on lipids, and this lipid is converted into fatty acid and glycerol. Then the enzyme trypsinogen, it is also present. This enzyme is also present in pancreatic juice. Pancreatic juice is also contains enterokinase. Trypsinogen, when it is present, it is in inactive form. For activation of trypsinogen, it must be converted into trypsin. And this trypsinogen, it is converted into trypsin in the presence of enzyme known as enterokinase. This reaction is also takes place in alkaline media. And now this trypsin acts on proteins, remaining part of protein, and peptones and proteases are produced in stomach by the action of pepsin enzyme. So this trypsin acts on protein, peptones and proteases, and these parts are converted into polypeptides. Other enzyme is also present in this pancreatic juice. It is known as chymotrypsinogen. This chymotrypsinogen, it is also present in inactive form. And for activation of this chymotrypsinogen, it must be converted into chymotrypsin. And this conversion takes place in the presence of trypsin. So here reaction chymotrypsinogen, which is present in inactive form, it is converted into chymotrypsin, which is active form. And this reaction takes place in the presence of trypsin. And now this chymotrypsin, it acts on polypeptides. And these polypeptides, in the presence of chymotrypsin, these are converted into dipeptides. Then in pancreatic juice, nucleases are also present. Nucleases, nucleotides, nucleosides, these enzymes are also present. And these enzymes are helpful for conversion of nucleic acid into sugar and base. At first, nucleic acid is converted into nucleotides in the presence of nucleases, and then this nucleotide is converted into nucleosides in the presence of enzyme nucleotidase, and this nucleoside lastly converted into sugar and base in the presence of enzyme nucleosidase. This is these are the enzymes. So you remember that in pancreatic juice, pancreatic amylase is present, pancreatic lipase, then enterokinase, chymotrypsinogen, trypsinogen, then nucleases, nucleotides, and nucleosides. These enzymes are present in pancreatic juice. And in pancreatic juice, maltose is formed. Then fats are converted into fatty acid and glycerol. Then the part of protein known as enzyme trypsinogen is converted into trypsin, and it helps for the digestion of protein. And here polypeptides are. And polypeptides in the presence of chymotrypsin are converted into dipeptides. So in this place, that is in intestine, the proteins are finally converted into dipeptides. This is the digestion which takes place in intestine. Then here third intestinal wall also secretes the enzyme, and this intestinal secretion of intestine is known as intestinal juice, or it is also known as succus syntheticus. Here always question is asked, intestinal juice, the another name for intestinal juice, that is succus syntheticus. Now in intestinal juice, the walls secrete the intestinal juice, and in intestinal juice also different enzymes are present. These enzymes are maltase, sucrase, lactase, dipeptidase, and lipase. These enzymes converts At first here, enterokinase convert trypsinogen to trypsin chymotrypsinogen to chymotrypsin. हे आपण मागच्या रिएक्शनमध्ये बघितले. Now this maltose, it adds on maltase enzyme adds on maltose and maltose is converted into glucose. Then sucrase enzyme adds on sucrose and it is converted into glucose and fructose. It is simple sugar. And lactase enzyme acts on lactose and it is converted into glucose and galactose. In this way, carbohydrates or starch, it is completely converted into simple sugar, that is glucose, fructose and galactose. Now about protein. Due to this pancreatic juice, dipeptides are formed. Means proteins are converted into dipeptides. And the enzyme dipeptidase, here dipeptides are converted into amino acids. In this way, here digestion of protein is completed, that is proteins are converted into amino acids. And emulsification of fat takes place due to bile salts. And these emulsified fats are converted into fatty acid and glycerol in the presence of enzyme lactase. In this way, the digestion of carbohydrate takes place, protein and fats takes place. After digestion of carbohydrate or starch, it is converted into simple sugar, that is glucose, fructose and galactose, while proteins are converted into amino acids and fats are converted into fatty acid and glycerol. At the end of digestion, chyme हा प्रोडक्ट तिथे तयार झालेला होता स्टमकमध्ये लास्ट. This chyme is converted into liquid form and this liquid form of chyme, now it is known as chyme, and it contains all the nutrients which are converted into simple form, that is simple sugar, amino acid, fat and glycerol. Next part is absorption, assimilation and digestion. Absorption means the digested food material is absorbed in blood vessels. Intestines are covered by number of blood capillary blood vessels, and this digested food material which is present in simple form, it is absorbed in this blood vessel. Absorption of glucose, amino acid, fatty acid, electrolytes, mineral, water takes place in various parts of alimentary canal. In lymphatic vessels, the fat globules are absorbed and these are known as lacteals. Then here assimilation takes place. Assimilation, मराठीमध्ये त्याला सात्विकरण म्हणतात. Assimilation means the absorbed food material, it becomes a part of protoplasm. It forms here nucleus and cytoplasm. This is known as assimilation. And last part, from this digestion, from this process, it is digestion. Digestion means undigested food material is converted into fecal matter. It reaches into the colon and rectum and it is given out, or its digestion takes place through anal.